Assalamualaikum dear students. I hope you all are good. Earlier we have completed the process of photosynthesis. Now today we'll be starting with the respiration. You might have already read in your 10th standard, 12th standard what is respiration. It is the breakdown of the energy rich molecules, which can be carbohydrates, which can be fats, which can be lipids. Fine. So here we are taking into account the glucose molecule here. Respiration will be studying and will be taking the glucose molecule in account. Here you see when glucose breaks down in presence of oxygen, it releases carbon dioxide plus water and lot of energy in the form of ATP, and this process is called as respiration. And the opposite of this, you can see here, when carbon dioxide plus water combine, they give glucose plus oxygen. Fine. So this is the respiration and this is photosynthesis. Now we are going to start with the respiration. Here is the overall reaction of the respiration. You can see when one molecule of glucose combines with 6 molecules of oxygen, it liberates 6 molecules of carbon dioxide plus 6 molecules of water and energy in the form of ATP. Fine. So this is the overall reaction of the this respiration, and it has been seen that the complete oxidation or complete degradation of the glucose molecule releases 30 or 32 ATP. Fine. A cell makes 30 or 32 ATP when glucose is degraded to carbon dioxide and water by respiration. Fine. So moving next, let me tell you, respiration is of two types. The first type is aerobic respiration. Fine. It takes place in the presence of oxygen. And number second is anaerobic respiration. It takes in the absence of oxygen. What is aerobic respiration? It takes place in presence of oxygen, and it is the complete oxidation. Glucose is completely oxidized. Fine. And it liberates pyruvate at the end. Fine. So here the energy produced is much more than this, but here you can see the anaerobic respiration. It is the incomplete oxidation of the respiratory substrates. By respiratory substrates here I mean glucose. Fine. So it produces less energy as compared to the aerobic respiration. Fine. So moving to the next, aerobic respiration, which takes place in presence of oxygen, is a set of enzyme catalyzed reactions, you can say, and these reactions are divided, or can be grouped into three major processes. Number one is glycolysis. Fine. And it takes place in the cytosol of all living organisms. Glycolysis takes place in the cytosol of all living organisms. Next is strict acid cycle or Krebs cycle. It takes place in the mitochondrial matrix. जो mitochondria होता है उसका जो matrix होता है, like in chloroplast we have stroma, same we have mitochondria. In mitochondria we have matrix, and it takes place in the mitochondrial matrix of eukaryotic cells and in the prokaryotic cells like bacteria. They don't have any mitochondria and organelle differentiation. So where does citric acid cycle take place in prokaryotes? It takes place in the cytosol. Fine. And oxidative phosphorylation is the third major process of respiration. Aerobic respiration, you can say, it takes place in the mitochondrial membranes of eukaryotes, and in prokaryotes it takes place in the plasma membrane. So moving ahead, we are starting with the first set of reactions, that is called the glycolysis or embedded Meyerhof pathway, EMP. It's also called as EMP or glycolysis. So why it's called as glycolysis? Here you can see glyco here means glucose, any sugar, and lysis means breakdown. Fine. The lysis means breakdown. So it is the breakdown of the glucose. Fine. And breakdown of glucose into 2-3 carbon molecules called pyruvates. Fine. Here pyruvates are released, and the glycolysis does not, this phase of respiration does not require oxygen. So the glycolysis takes place in anaerobic organisms also, as it does not involve any use of oxygen. So it takes place in the anaerobic organisms also. So here is the glycolysis. It has two phases. Number one is preparatory phase, and number second is payoff phase. It total has total has 10 steps. Here you can see I have marked. It has 10 steps. 5 steps. The first 5 steps are preparatory phase, and the second, from step 6 to step 10 include the payoff phase. So what here is done, we have a glucose molecule. Here you can see we have a ring structure of the glucose molecule. What happens in presence of ATP? When ATP gets converted to adenosine triphosphate, means triphosphate, it has 3 phosphates, it releases adenosine diphosphate plus a phosphate group in presence of enzyme hexokinase. Please remember, in presence of hexokinase, it releases what? It forms what? Glucose, but 6 phosphate. Here the branch chain structure of the glucose is formed, but the phosphate released from ATP gets attached at the 6th carbon. Here you can see the phosphate group released from the ATP gets attached at the 6th carbon. Here you can see. And now what happens, this glucose 6 phosphate, as it has phosphate group at the 6th carbon, it gets phosphoglucoisomerase. In presence of the enzyme phosphoglucoisomerase, it changes into fructose 6 phosphate. It gets isomerized, you can say, and the enzyme is phosphoglucoisomerase. And what happens here, it changes into fructose 6 phosphate. Now this is not glucose anymore, this is fructose. Here you can see the configuration, and the phosphate group remains at the 6th carbon only. Fine. Here this phosphate comes from the ATP here. Please remember this. Now what happens, another ATP is used here. The ATP gets converted to ADP here. Fine. And the enzyme used here is phosphofructokinase. Phosphofructokinase. Now what it does, it adds another phosphate which is released from the ATP here, and now this fructose 6 phosphate becomes fructose 1,6 phosphate. It already has phosphate at the 6th carbon, and the new phosphate released from this ATP gets attached at the 1st carbon. Now it becomes fructose 1,6 bisphosphate. Here you can see it has 2 phosphates which have come from the ATPs. Now this, what happens, this fructose 1,6 bisphosphate changes into glyceraldehyde 3 phosphate. It is a 3 carbon compound. Here is, you can see it has 3 carbons, glyceraldehyde 3 phosphate, and it has the phosphate group at the 3rd position. And what happens, this in the step 5, it gets changed into, it breaks down. Here you can see the fructose 1,6 phosphate breaks into 3 carbon compound. Here it's 3 carbon and it's 3 carbon dihydroxyacetone phosphate. Dihydroxyacetone phosphate. It also has a phosphate. Fine. So here, and these dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, they keep on interchanging. That is with the help of enzyme triose phosphate isomerase, or they get isomerized. They are isomers of each other, and they get isomerized in the presence of enzyme triose. Triose here means because it converts the 3 carbon compounds. Triose means it converts, it acts upon the 3 carbon sugars here and interchanges them. Fine. Now what happens, now 2 molecules of this here 2 times. 2x means 2 times. 2 times glyceraldehyde 3 phosphate changes into 1,3 bisphosphoglycerate. Fine. Here 2 molecules I have, so it changes into 1,3 bisphosphoglycerate. So what happens here, this NADP positive plus phosphate gets changed into NADH plus H positive. Now here it also gives it a phosphate, and this phosphate gets attached here and it becomes 1,3 bisphosphoglycerate. I have this compound here, and now what happens here, 1,3 bisphosphoglycerate, it gets changed to 3 phosphoglycerate, means it has 2 phosphates here at carbon 1 and carbon 3. Now one of the phosphates is removed, and what happens to that phosphate, it is utilized by here by the ADP. यहाँ पे ये ADP use करता है इसको, and it gets changed into ATP. So we get a molecule of ATP here. So here we are taking 2 molecules. So here 2 ADPs change into 2 ATP. Fine. So ATPs are formed in this phase, and it takes place in presence of the enzyme phosphoglycerate kinase. And now what is the next step? This 3-phosphoglycerate, it gets converted to 2-phosphoglycerate. Here, this phosphate is at the third carbon. Now the phosphate gets attached at the second carbon. That is why it becomes 2-phosphoglycerate. It becomes 2-phosphoglycerate. In the presence of the enzyme phosphoglycerate mutase. In the presence of enzyme phosphate glycerate mutase. And what happens here? Water is released. Water is taken out in the presence of enolase. So the reaction in which water is released are called a dehydration. Here a dehydration reaction takes place, and as a result what happens? It changes into phosphoenol pyruvate. Phosphoenol pyruvate. You have already read in the C4 cycle, there is a this phosphoenol pyruvate PEP, which combines with the carbon dioxide to form oxaloacetic acid, is that phosphoenol pyruvate. It has a phosphate group attached at the second carbon. So what happens later on? This phosphate is now removed, and as a result what happens? This phosphate is utilized by this ADP and it gets converted to ATP. Fine. Again here 2 times means 2 ATP were released here and 2 ATP were released here. So 2 ATP are formed. So अगर हम इनको बैलेंस करेंगे, you later on see the detailed account of this. यहाँ पे 2 ATPs बनते हैं, यहाँ पे 2 ATPs बनते हैं. And as a result of this, in the presence of enzyme pyruvate kinase, what happens? This phosphoenol pyruvate, it becomes pyruvate. It becomes pyruvate. It has no phosphates. Phosphates are consumed by the ADPs and change into ATP. So this is the payoff phase. In payoff phase, we get the energy in the form of ATP. In the form of ATP. So here you see 4 ATP are formed and 2 NADH are formed in the payoff phase. Please remember, 4 ATP and 2 NADH are formed in the payoff phase of the glycolysis, and in the preparatory phase 2 ATPs are consumed, because here glucose is here, we don't have any phosphate, the ATPs give phosphate to it and it becomes glucose 6-phosphate. The phosphates come here from the ATP. Fine. So 2 ATPs are consumed in the preparatory phase and 4 ATP are formed in the payoff phase. So the number of ATP, अगर हम टोटल नंबर ऑफ ATP लें, अब 2 हमें कंज्यूम हुए हैं, 4 हमें बने हैं. सो नेट प्रोडक्शन इज 2 मॉलिक्यूल्स ऑफ ATP. अगर हम 4 - 2 करेंगे तो 2 आएगा, एंड what happens? 2 molecules of NADH. So this is the overview of the glycolysis. I hope I'm clear with this. So if there are any questions, please let me know in the comments. I'll be replying to you. Thank you so much. Stay blessed, stay safe. Tomorrow we'll be starting with the Krebs cycle, inshallah.